Hi, welcome to part 3 of this lecture, and in this part 3 we are going to discuss about anaerobic respiration. Do you know what is anaerobic respiration and what is the difference between anaerobic respiration and also aerobic respiration? Now, before we go further into anaerobic respiration, at this point we should know that glucose is not the only material that can be oxidized to release energy, which other than glucose, lipids and proteins can also be broken down and be used for oxidation to generate ATP. So please read further in which between lipids and proteins which will produce high energy and why. What does it mean by anaerobic respiration? Anaerobic respiration is a respiration without oxygen. Oxygen isn't around to act as an acceptor at the end of electron transport chain. Some prokaryotes can live in low oxygen environments in which they will rely on anaerobic respiration to produce ATP. Some of them they will use sulfate or other inorganic molecule as final electron acceptor other than oxygen at the end of electron transport chain. Fermentation is another anaerobic pathway for breaking down glucose, and it involves only glycolysis as the energy extraction pathway. In the absence of oxygen, pyruvate from glycolysis does not enter mitochondria. Fermentation uses substrate-level phosphorylation instead of oxidative phosphorylation to generate ATP. Two common types of fermentation are alcohol fermentation and also lactic acid fermentation. In alcoholic fermentation, pyruvate produced from glycolysis will not enter mitochondria. This is due to the absence of oxygen. Alcoholic fermentation can happen in plant and in fungi. In glycolysis, one molecule of glucose will be broken down to two molecules of pyruvate. Each pyruvate has 3 carbons in their structure. 2 ATP are also produced in glycolysis. The ATP produced here is by using substrate-level phosphorylation. NAD+ will be reduced to NADH in glycolysis. The pyruvate at the end of glycolysis will next undergo decarboxylation into 2-acetaldehyde. Each of these acetaldehyde, also called as ethanol, will have only 2 carbons in their structure. Next, these 2 acetaldehyde will be reduced by NADH to produce 2-ethanol. So by the end of alcoholic fermentation, from one molecule of glucose there will be 2 molecules of ethanol. The products of anaerobic respiration are toxic, and if this happens in yeast, the alcohol accumulation will rise to a level where until further fermentation is prevented, and this will kill the yeast. This alcoholic fermentation is also non-reversible. Next pathway is lactate fermentation. In lactate fermentation, the pathway can be reversed in mammals. Meaning here is that when there is oxygen available again, the lactate can be oxidized to form carbon dioxide and water. In lactate fermentation, there are no intermediates being produced. The pyruvate produced from glycolysis will be directly reduced to 2-lactate by NADH, and 2-ATP is produced from glycolysis only, and this ATP is of course produced by using substrate-level phosphorylation. The lactate here can be broken down to release energy if oxygen is available again. Mussels carry out lactate fermentation only during extreme exercise. When this happens, the lactate produced will accumulate and this can cause muscle cramp. There are various applications of anaerobic respiration. Based on alcoholic fermentation, first one, bread can be made. The carbon dioxide released from the reaction will help to raise the dough. Second, tapai can be made by alcoholic fermentation, in which cooked white rice is washed, cooked and cooled, and then they are mixed with yeast. The tapai that is formed will taste sweet and contain a little alcohol. Based on lactate fermentation, yogurt can be made by adding a suitable bacteria inside the milk and incubating them together for a certain period of time. And then the enzyme lactase from the bacteria will act on the lactose to produce lactic acid or the lactate. This lactic acid will curdle the milk and give it a sour taste. We have finally reached the end of this chapter. If you have any question regarding on this chapter, please post your question in the forum section in your Smart B3 VMS. When you want to study for this chapter, please refer back your chapter learning outcomes. So I bid you all good luck for your exam and thank you.